So this is the fourth problem solving session for the course Biochemistry. The course instructor is Professor Swagata Dasgupta. You might have seen her videos till now. So I'm Shrija Ghosh, a PMRF from IIT Kanpur, and I'll be taking the problem solving session. So today's session is about enzymes. So we'll study about the kinetics of enzyme that it follows, and we'll solve the questions related to that topic. And if anybody has any doubt at any point, you can stop me and ask, or you can write it in the chat box and I'll try to clear your doubts. We'll solve the questions that are like basically we'll solve the questions related to your assignment that has been given to you for the fourth week videos. So let's start the session now. So starting with the first question, which of the following best describes the assumptions made in steady state kinetic analysis? First statement is the concentration of S, that is the substrate, is decreasing. Second statement is the concentration of ES, that is the enzyme substrate complex, is constant. Third statement is the total amount of enzyme decreases. So which of the following best describes the assumptions made in the steady state kinetic analysis? So anybody wants to answer? If anybody knows the answer, they can write in the chat box, or unmute and you can give the answer. No one? So what is steady state kinetics? We are using the term steady state kinetic analysis. So what do you mean by a steady state? Steady state means any reaction, if it is at a steady state, that means the rate of formation of some compound versus the rate of breakdown of that compound, both the rates are equal. So it's a steady state that has been maintained. So in this case, if you see, the correct answer is C, that is the second statement is correct. Why? The concentration of ES is constant. So we will see here, if you see the reaction that is being followed by an enzyme and a substrate. So what an enzyme does, it can convert a substrate into product. Okay, basic enzyme reaction is mainly you have a substrate, the substrate binds to the enzyme and then it converts into product, and the product is released and the enzyme is free again for another substrate molecule to bind to it. Okay, that is the basic enzyme substrate reaction. Now in this reaction, if you see, once the enzyme binds to the substrate, it forms the intermediate, that is the enzyme substrate complex, ES. We denote it by ES. So when that ES is formed, the ES has two fates. Either the ES can again dissociate into the enzyme and the substrate, or the ES can get, the substrate can get into the get converted into the product and get released from the enzyme, and we will get the free enzyme. So basically, let's say you have an enzyme molecule with a substrate pocket. So our substrate will fit into this pocket, and this is the enzyme. So this is the enzyme substrate complex. After this, it can again dissociate back into the free enzyme and the substrate molecule, or it can move forward where we will get the free enzyme and we will get the product molecule, which will be different from the substrate, and it will dissociate from the enzyme. Okay, so now we are saying at steady state the rate of formation of ES, that is E plus S giving rise to ES. The rate of formation of ES is equal to the rate of breakdown of ES. Then what do you mean by that? That means any small change in ES concentration at a given time will be 0. Okay. So that means dES by dt will be 0. And how can we show the change in the ES concentration? The change in the ES concentration would be formation of ES subtracted by the breakdown of ES. So formation of ES, how can you what is the rate constant for formation of ES? E plus S goes to ES, that is the rate constant is K1. Okay. So K1 into the reactant concentration. So here the reactants are E and S. So concentration of E multiplied by concentration of S. So this is the formation. This is what? This is formation. Okay. Now we will see the breakdown. So this term and this term, there are two terms for breakdown. Breakdown of ES. Why there are two terms? Because the ES can either break down into E plus S again, that is the reverse reaction of this equilibrium, or ES can move forward for and become E plus P. Okay. So for the backward reaction we have the rate constant K minus 1 and the dES by dt, the change in the ES concentration will be determined by negative of K minus 1 into ES concentration. Why negative? Negative sign is there because it is the breakdown. For formation we have positive sign, for breakdown we have negative sign. So K minus 1 into ES. And again there is another breakdown step, that and the rate constant is K2. So K2 into ES. So minus K minus 1 into ES and then minus K2 into ES. The whole thing is equal to 0. Why 0? Because it is at steady state. So that means at steady state we have the formation of ES is equal to breakdown of ES. That means the concentration of ES is constant at any given time. This is clear? So at any given time the concentration of ES is constant. Okay. So that is the first assumption that was made in steady state kinetic analysis. So whenever we write an enzyme substrate reaction, we assume that the ES, the kinetics for the formation and breakdown of ES, that is constant. Okay. So this is one assumption that was made for deriving the enzyme kinetic equation. Okay. We will go through the enzyme kinetic equation again like in the further questions. Prajakta, do you have a question? You were asking for the recording for this session, right? Prajakta? Yes, Prajakta, you can unmute and ask. Okay. Okay, so moving to the next question. This question is clear, I hope. If any one of you has any doubt, you can either write in the chat box or you can unmute and ask. So the next question. Question number 2. The class of enzyme that catalyzes synthetic reactions where two molecules are joined together and ATP is used is option A hydrolases, option B lyases, option C ligases, and option D isomerases. So which of the following class of enzyme catalyzes the synthetic reaction where two molecules are joined together with the help of ATP? Anybody knows the answer? Yes. Like Surbhi has answered ligases. So that is the correct answer. Option C ligases. So we have in total 6 classes of enzyme. Broad classification of enzyme has been done into 6 parts. So by the name of the enzymes only you can understand what are the functions of the enzyme. So the first class is oxidoreductases. They what they do? Oxidoreductases, they basically catalyzes all the redox reactions. So whether it's a oxidation or a reduction happening, yeah. So oxidoreductases basically catalyzes the oxidation or the reduction reactions. So all the enzymes that can basically act on many chemical groups to add or remove hydrogen atoms, those enzymes are classified under the oxidoreductase class. Okay. The second class is transferases. Now the name, by the name itself you can see transfer. Its role is to transfer functional groups between the donor or the acceptor molecule. So transfer of any functional group will be done by the enzymes that are classified under transferases. Okay. So one example of transferases is kinases. Kinases are special kinds of transferases that can transfer phosphate groups, okay, across the different molecules. Okay. Then the third class is hydrolases. Hydrolases is basically any enzyme that can catalyze hydrolysis. What do you mean by hydrolysis? Hydrolysis means addition of water across the bond. So if I have a peptide, you have studied about peptides in the past 3 classes. So if you have peptide bond is basically the bond between the amino and the carboxylic group of 2 amino acid. So hydrolysis of peptide bond will be the breakdown of the peptide bond by the addition of a water molecule. Okay. So that is hydrolysis. So any enzyme that can do this hydrolysis will be classified under hydrolases. Then comes lyases. So lyases, basically what they do, they can add water, ammonia or carbon dioxide across the double bonds or remove these elements to produce double bonds. So what can lyase do? Lyase can basically add groups across double bonds. So it is kind of between transferases and hydrolases, because it is transferring some group, but it is transferring across a double bond instead of a donor or acceptor molecule. Okay. And then hydrolases adds water, but hydrolases can add water across a single bond, whereas lyases can add water molecules across double bonds, or lyases can also remove water molecule to produce a double bond. Okay. So that is lyases. Any enzyme that can do this activity, this function, are classified under the group lyases. Then we have isomerases. What do you mean by isomers? Anybody, what do you mean by isomers? So isomers is basically any two molecules which has the same chemical formula but different structures. So DL isomers, and there are other kind of so you know the amino acid leucine and isoleucine. If you see the molecular formula of both leucine and isoleucine, you will see the molecular formula is exactly same, but the structure is different. Okay, so any two molecules which has the same chemical formula but different structure are called isomers. And an enzyme that can convert one isomer into the other is called isomerase. Okay, any enzyme that can convert one isomer into the other isomer. So basically any enzyme that can convert an L amino acid into D amino acid. Okay, so that enzyme would be called isomerase. Okay, then comes the last class, ligases, that basically catalyzes the reaction in which two chemical groups are joined. So ligase, what it does, it acts as a glue. It can help sticking two molecules together with the use of energy. From where the enzyme will get energy. Energy will be achieved from ATP molecule. Okay, so our question was the class of enzyme that catalyzes the reaction where two molecules are joined together using ATP. It is ligases. So ligases, if we have two DNA molecules and I want to stick them together, I want to make a single molecule using those two DNA molecules, I can use ligase, which will use an ATP and stick them together. Okay, so these are the 6 classes of enzymes with different broad functions. So based on the functions, the enzymes have been classified into 6 different groups. Any doubt till now? Prajakta, if you are having difficulty using the portal, I would suggest you can just connect with any of your batchmates and discuss. I hope you know the portal from where you are getting all the videos for the all the lecture videos, right? Lecture videos that have been posted by Professor Swagata. If you would have seen that portal, on the left hand side you will see week 1, week 2, week 3, week 4 written. Below that only you will see a tab saying problem solving session. You can find the link of the Excel sheet in that, and on that sheet all the YouTube links are posted for the recording of each session, and the slides are also shared there. Okay, so moving to the next question, question number 3. Number of substrate molecules are converted.
Hello, ma'am.
Yeah.
Hello.
Yeah, tell me.
हाँ मैम, एक्चुअली में क्या हम लेक्चर में होते हैं ना कॉलेज के, तो रिकॉर्डिंग वगैरह मिलती है सेशंस की डाउट.
Sorry, but there's a lot of disturbance. Can you be more clear?
ओके मैम, वेट फॉर 1 minute.
You will get the recording for each session on the portal only. Okay, the YouTube links have been shared. You can just go through the link, and all the recordings are posted on YouTube, so you can find it. Okay, so question number 3. Number of substrate molecules converted to product in a given unit of time by a single enzyme molecule at saturation is referred to as the option A, dissociation constant, option B, half saturation constant, option C, Michaelis-Menten number, and option D, turnover number. So the number of substrate molecules converted to product in a given unit of time by a single enzyme molecule at saturation is referred to as. Anybody knows the answer? Option B. Surbhi has answered option B. Anyone else? So the correct answer is actually option D, turnover number. So what is turnover number? Turnover number basically means the number of substrate molecules turned over per enzyme molecule. So if I have one enzyme molecule, okay, in each second or in each unit time, so if we are considering the time in seconds, then per second, or if we are considering the time in minutes, then per minute. So per unit time, how many substrate molecules can be converted into product by one molecule of enzyme? Okay, so if I have a molecule of enzyme, okay, let's say this is the enzyme. If I have a molecule of enzyme, so what will happen? A substrate molecule will come and get converted into product molecule. So this cycle will continue. One substrate molecule will come, convert into product molecule, then another substrate molecule will come and get converted into product molecule, and so on. So in one unit of time, like in 1 second or 1 minute, how many substrate molecules can enter the enzyme and get converted into product? So how many substrate molecules are turned over per enzyme molecule? So by one enzyme molecule, how many substrate molecules can be converted into product in 1 unit time is the turnover number. Is that clear? So turnover number is actually denoted by the term Kcat. So it basically gives the it can be used to determine the catalytic efficiency of the enzyme. So Kcat is basically equivalent to the rate constant of the rate determining step. So now this is the basic enzyme kinetic equation that we write. I have shown you this in the previous slide also. So enzyme and substrate combines together to form enzyme substrate complex, which eventually breaks down into enzyme and product. Okay. So in this whole step we have three rate constants. One is the enzyme substrate complex formation, and then there is one constant that that gives the breakdown of enzyme substrate complex back to enzyme and substrate, and the third where the enzyme is actually catalyzing the reaction and converting the substrate into the product and we are getting the free enzyme. So in all these three steps we have 1, 2 and 3 step, right? So among these 3 steps, which of the following is the slowest step? The slowest step is basically this, where the substrate is getting converted into product, because here the chemical reaction is happening. So enzyme, when it's bound to the substrate, now the substrate is getting converted into the product. So enzyme is actually catalyzing the reaction. The actual chemical reaction is happening. So this is the slowest step of this reaction. And since this is the slowest step of this reaction, it is called the rate determining step. So whichever step will be the slowest would determine the complete rate of the reaction. Okay. Okay, this is clear. So if I have this as the rate determining step, then Kcat will be equal to the rate constant of this rate determining step, that is K2. Okay, and also we know that the rate.
Hello.
Yeah.
Yeah, ma'am. Should I talk?
Yeah, tell me.
यस, एक्चुअली मुझे ये पूछना था मैम कि अगर हम ये डाउट सेशन में अगर नहीं अटेंड कर पाए ड्यू टू माय कॉलेज लेक्चर वगैरह, तो इसकी लिंक वगैरह किधर मिलेगी, मीन्स रिकॉर्डेड?
I told you na, the links are there on the portal, NPTEL portal, where you see the weekly videos. Have you seen any of the weekly videos, week 1, week 2 by Swagata ma'am?
यस, यस,
Yes. So on
यस।
that same portal, on the left hand side, you might have seen the tab called problem solving session. Okay, on that
ओके,
same portal, on that same page. Okay, you will see
ओके,
a tab saying problem solving session. Click on that. You will find a link. Click on that link. There will be an Excel sheet with all the YouTube links pasted there. You can just click on each YouTube link for each week's video each week's session, problem solving sessions. Okay.
ओके, थैंक यू सो मच मैम।
Yeah, welcome.
यस।
Yeah, so where was I? Yeah, so the turnover number that is denoted by k cat is equivalent to the rate constant of the slowest step, that is k2. And now since this is the rate determining step, the rate of the equation that is given by V can be written as k2 into the ES concentration of the enzyme substrate complex, because this is the slowest step, and we can write the rate as the rate constant multiplied by the concentration of the reactants. Okay. Now here we know that the total enzyme concentration ET is equivalent to concentration of free enzyme plus concentration of the enzyme substrate complex, whatever enzyme I am adding into the reaction. Let's say I have added 1 mole of enzyme, okay, or 1 molar of enzyme. So whatever concentration I am adding, either that enzyme can be free, or that enzyme can be in the bound form with the enzyme, with the substrate. Okay. So total enzyme concentration will be concentration of free enzyme plus concentration of enzyme substrate complex. Now when all the molecules are all the enzyme molecules are saturated with enzyme with substrate, because if you see in the question we have at saturation, there is a word called saturation. What do you mean by saturation? That all the enzyme that is present in the system have been saturated with the substrate. So let's say there are 4 molecules of enzyme, all 4 molecules are now bound with substrate. So there is enough substrate present, enough substrate molecules present in the solution that all the enzyme molecules are completely saturated. There is no free enzyme present in the system. If there is no free enzyme present in the system, the total enzyme concentration will be equal to the enzyme substrate complex, and in that case we can say the rate of the reaction is maximum, because there is no more free enzyme that can bind to any more substrate and catalyze the reaction. So that rate will be maximum, and that maximum rate can be written as k cat into the total enzyme concentration. So V max, that is the maximum rate of the reaction, is equivalent to the turnover number of the enzyme multiplied by the total enzyme concentration that we have added in the reaction. Okay. So turnover number is a very important term that we need to calculate if we want to know the efficiency of the enzyme, how efficient our enzyme is, how fast our enzyme molecule can convert the substrate into product. Okay. I hope this is clear. What is turnover number? So we will move to the next question, that is question number 4. Which of the following statement is true about the region of the graph below where concentration of substrate is very less than km? Okay. So now we have here a typical Michaelis-Menten curve. Okay. And they have asked which of the following statement is true about the region where the graph is graph below where we have the substrate concentration as very low, less than Km. That means at somewhere near the origin where the substrate concentration is very less, we have to say which of the following statement is true about this particular region. Okay. So now if we see the statements for this part of the graph, concentration of enzyme will be very greater than concentration of substrate. Then second statement is when substrate concentration is very less than Km, the initial rate is equal to catalytic efficiency multiplied by the free enzyme and substrate concentrations. Then third statement is when substrate concentration is very less than Km, the rate is zero order with respect to substrate. Then the fourth statement is this region allows for the determination of Km. Okay. So let's see what will be the correct answer. Anybody knows the answer or wants to guess? So the correct answer for this question is option B, and we'll see how. So this is a Michaelis-Menten curve, typical Michaelis-Menten curve. So we know the Michaelis-Menten equation as rate of the reaction, that is V, is equal to V max multiplied by the substrate concentration S divided by Km plus S. Okay. So this is the enzyme substrate reaction where the Michaelis-Menten equation states V is equal to V max S plus Km plus S. Okay, V max S divided by Km plus S. Now we are saying when substrate concentration is very less than the Km. So if substrate concentration is very less than Km, we can ignore the substrate term from the denominator, because let's say the Km is 1 millimolar and substrate concentration is 10 raised to the power minus 5 millimolar. Okay. So in that case 1 plus 10 raised to the power minus 5 won't make a very significant difference. It will be almost equal to 1 only. So we can rewrite the Michaelis-Menten equation as V is equal to V max upon Km multiplied by S, concentration of substrate. Now if you look at this reaction, this is called a first order reaction. So if you look at this reaction, sorry, just a second. So if you look at this reaction, V is directly proportional to the substrate concentration. So this is called first order reaction. So if you see the options, option C was saying rate is zero order with respect to S. So this is not true because it is a first order reaction. Okay. Now for a first order reaction where rate of reaction is directly proportional to the substrate concentration, we have in the previous slide only I have explained you why Vmax is equal to Kcat into enzyme concentration, right? So now if I replace the value of Vmax by Kcat into enzyme concentration, we will get Kcat into enzyme concentration multiplied by substrate concentration divided by Km. Now this Kcat by Km is actually called the catalytic efficiency of the enzyme. So I was telling you that Kcat or the turnover number can be used to determine the catalytic efficiency of the enzyme. So if you divide the Kcat by the Michaelis-Menten constant, that is Km, you will get the catalytic efficiency of enzyme. How? Why? Because Km actually can tell about the binding efficiency of the enzyme. So if we have a high Km, the binding will be low. If we have a low Km, the binding will be high. Okay. And if we have high Kcat and low Km, the Kcat by Km ratio would be high. That will give the higher catalytic efficiency. So we can actually tell the catalytic efficiency of an enzyme by determining this ratio kcat by km. Okay, and if you see now in the option we had initial rate is equal to the catalytic efficiency multiplied by the free enzyme and substrate concentration. So if you see, if this is the catalytic efficiency, so rate is equal to catalytic efficiency multiplied by the enzyme and substrate concentrations. Okay, so that's why option number B is the correct answer. So at the starting part, so when you have substrate concentration less than Km, so this is the region we are talking about. So this part is almost linear and follows a first order kinetics. This is clear? This is a very important part. So there's a condition where substrate concentration is very less than Km. Now we will look into the second case where the substrate concentration is very higher than Km. Okay, let's see the next question. Question number 5. Which of the following statement is true about the region of the graph where the substrate concentration is very higher than the Km? So now we are talking about this region. Okay, when the substrate concentration has exceeded the Km value. Okay, so anybody wants to answer based on the previous question? If anyone can tell me the answer for this question. I told you how to find out the answer for the previous question, so if you can solve based on that same concept. So we have the Michaelis-Menten equation, V is equal to V max S upon Km plus S. Okay, so now if I have the substrate concentration higher than the Km, significantly higher than the Km, then from the denominator we can actually eliminate the term Km. What will happen if we do that? Anyone wants to answer? Anybody knows the answer? So now if you see here, if I eliminate the term Km, so we will have V max S divided by S. So we can actually cancel out these terms and we will get V is equal to V max. So what do you mean? So when we have substrate concentration significantly higher than Km, the maximum velocity will be attained by the reaction. So this means the velocity is not dependent on the substrate concentration at all. That is, the rate is zero order with respect to substrate concentration. So if at this stage now I add more and more substrate to the reaction, what will happen? There will be no change in the velocity because now the enzyme has reached its maximum velocity. So even if you keep on adding substrate after this, there will be a saturation and there won't be any more increase in the velocity after Vmax. So this point in this curve, so this point is basically the Vmax of the reaction, sorry. So this is Vmax. So an enzyme reaction will not exceed Vmax. So once it achieves Vmax, after that you keep on adding more and more substrate, there won't be any further increase in the reaction rate. Okay, so the reaction rate is zero order with respect to the substrate concentration. Okay, rate is independent of the concentration of substrate, that is, the rate becomes zero order with respect to substrate. This is clear? So there are two conditions basically, one where the substrate concentration is very less than Km. The second case is substrate concentration very higher than Km. There is a third case where the substrate concentration is equal to Km. We will look into that in the upcoming questions. Okay. So these three cases are very important when we are solving enzyme kinetic questions. Okay, because this is where we can see. So if I have a actual example of a, like if I see someone has set up a reaction where they have taken the substrate concentration very higher than the Km of that enzyme. So I'll know that the enzyme is going to follow a zero order reaction. That is, the enzyme will work at its maximum capacity. Okay. And if I can see that the person has used the substrate concentration very lower than the Km, like they have used the substrate concentration that is very low than the Km value for that particular enzyme, I can say the reaction will follow first order kinetics. That is, it will be directly proportional to the substrate concentration. So if I at that time, if I add more substrate, it will directly influence the velocity of the reaction. This is clear? So these two cases are very important. So moving to the next question, question number 6. Michaelis and Menton assumed that the overall reaction for an enzyme catalyzed reaction could be written as E plus S gives ES, that is the enzyme substrate complex, that breaks down into E and P. P is the product. Using this reaction, the rate of breakdown of the enzyme substrate complex can be described by the expression. Option A is K1 multiplied by the total enzyme concentration plus the enzyme substrate complex. Option B is K minus 1 multiplied by the enzyme substrate complex plus K2 multiplied by the enzyme substrate complex. Option C is K1 multiplied by the total enzyme concentration plus enzyme substrate complex and then multiplied by the substrate concentration, and option D is K2 multiplied by the enzyme substrate complex concentration. So we have to tell the rate of breakdown of enzyme substrate complex. Okay. Anybody wants to answer? I have actually discussed a similar question in the previous slide. You should be able to answer this question. So we have here two steps where the enzyme substrate is breaking down. I have told you. So what are those two steps? Let's see. So here we have the enzyme substrate complex. So formation of enzyme substrate complex is given by the term K1. The rate constant for the formation of enzyme substrate constant is K1. Now the enzyme substrate complex is actually breaking down into two steps. One where the rate constant is K minus 1 and the enzyme substrate complex is dissociated into free enzyme and the substrate, that is the reverse reaction. Whereas the forward reaction where the rate constant is K2, we have the conversion of substrate into product and release of the free enzyme. So breakdown of ES, so breakdown we have two steps, right? So one is where the rate constant is K minus 1. The second is the rate constant as K2. Now, if I have to write the rate of the breakdown, so rate of any reaction, if I have a reaction A plus B goes to C given by K, if I have to write the rate of the reaction, the rate will be written as the rate constant multiplied by the concentration of each reactant. Okay. So here if I have to write the rate of breakdown, that is ES going to E plus S, the rate constant is K minus 1 and the reactant is ES. Okay. Because ES is going to E plus S. We will see individual reactions now. Okay. One reaction is this, second reaction is ES going to E plus P. Okay. This is the first reaction, this is the second reaction. So for the first reaction we have K minus 1 into ES. For the second reaction we have K2 into ES. This is clear? So option number B is the correct answer. Correct answer is option number B. This is clear? I hope this is clear. So basically breakdown is in two reactions, ES going to E plus S, where the rate constant is K minus 1. So the rate of that reaction is K minus 1 into the concentration of ES. Then the second reaction is ES going to E plus P, where the rate constant is K2, and the rate of the reaction will be written as K2 into concentration of ES. This is clear? Moving to the next question. Till now, if you have any doubt, you can ask me, you can unmute, or you can write in the chat box, okay? So next question is question number 7. Student conducts an experiment to test the efficiency of a certain enzyme, which would result in a change in the efficiency of the enzyme. Option 1 is adding more substrate but not enzyme. Option 2 is carrying out the experiment in absence of water, and option 3 is adding an acidic solution to the reaction mixture setup. So what do you think, which of the following would change the efficiency of enzyme? Anybody? Which of the following can change the efficiency of enzyme? Do you think adding more substrate can change the enzyme's efficiency? Or carrying out the experiment in absence of water can change the enzyme's efficiency? Option C. Carrying out the enzyme experiment in absence of water. Okay. Anyone else? So Karuna has answered option C. You don't think that adding an acidic solution to the reaction mixture setup can change the efficiency? If I add acid to a reaction mixture, what will happen? The pH will decrease, right? Okay, Vijay Lakshmi has answered option D. Okay, so let's see. The correct answer is option D. So let's start with the third statement only. So adding an acidic solution. So any enzyme, it follows a bell-shaped curve for pH and temperature based experiments. So basically, if I have a pH scale, and this is, let's say, the rate of reaction. So every enzyme will have an optimum pH where the rate will be maximum, and below and above that optimum pH the rate will change. Okay, now if the rate is changing, the enzyme efficiency will ultimately change, because I've told you how do we determine the enzyme efficiency. It is basically Kcat by Km. Okay, so Kcat will change, catalytic efficiency will change if I change the pH. If I decrease the pH, we are adding acidic solution, so we are decreasing the pH. If I decrease the pH, the efficiency will decrease. Okay, so enzyme efficiency will change by adding acid to the pH. Enzyme efficiency will change by if we carry out the experiment in absence of water. Why? Because now water, all the enzymes we know are proteins. Okay, and if we have a proteins generally like water environment. Okay, so if I have a protein, the catalytic sites are generally charged amino acids. So the catalysis will always happen in presence of water. So water is always a second substrate to most of the enzymes present in nature. So if we are carrying a experiment in absence of water, most likely the enzyme won't be efficient at all. So removing water will definitely affect the enzyme's efficiency. Third is adding more substrate but not enzyme. So if we are not adding more and more enzyme, and if we keep on adding substrate, what will happen? There will be a saturation. Okay, I talked about the saturation. So what will happen? The enzyme rate will reach the maximum velocity. Okay, so all three can actually change the enzyme's efficiency. Okay, I hope this is clear. Moving to the next question, question number 8. In a Lineweaver-Burke plot, a non-competitive inhibitor shows which of the following effects? First statement is it moves the entire curve to the right. Second statement is it moves the entire curve to the left. Third is it changes the y-intercept. Fourth is it has no effect on the slope. So in a Lineweaver-Burke plot, a non-competitive inhibitor shows which of the following effect? Anyone? If you have studied about Lineweaver-Burke plot for the inhibitor kinetics, anybody remembers how the plot looks? Which of the following option will be correct? Anyone? So the correct answer for this question is option D. Why option D? It changes the y-intercept. So we have majorly 4 type of inhibitors: competitive, uncompetitive, non-competitive, and then mixed inhibition. So mixed inhibition is basically in between of all three. So in case of competitive, uncompetitive, and non-competitive inhibition, you will get a typical kind of Lineweaver-Burke plot. Okay, so here in the question we are talking about non-competitive inhibition. So you see the Lineweaver-Burke plot for the uninhibited and inhibited will have the same x-intercept and different y-intercept and different slope. So slope and y-intercept will keep on changing, whereas the x-intercept will be same. So wherever the graph crosses the x-axis is called the x-intercept. Wherever the graph is crossing the y-axis is called the y-intercept. You see, in case of competitive inhibition, the y-intercept remains same and the x-intercept keeps on changing, and also the slope keeps on changing. For uncompetitive inhibitor, you will see both y and x-intercept keeps on changing, but what is constant here? The slope is constant. So these are parallel lines. So the uninhibited and the inhibited will have parallel lines, okay, on the graph. So the x and y intercept will be different, but the slope will be same for all the cases. So in case of uninhibited and inhibited reactions, the slope will be same. And what is the slope for a Lineweaver-Burke plot? It is basically Km upon Vmax. Okay. So we will see in the upcoming questions what is the value of these x and y intercept and the slope of the Lineweaver-Burke plot. Okay, and in case of mixed inhibition, there is no particular pattern that is followed. So for non-competitive inhibitor, the y-intercept changes. There is no change in the x-intercept. The curve does not entirely the curve is not entirely moving to right or left, so these are not the right answers, and it does have an effect on slope. So only third option is correct. It changes the y-intercept. Okay, so it changes the y-intercept. Next question, question number 9. Which among the following type of enzyme can add water, carbon dioxide, etc. to the double bond? Option A, isomerases. Option B, ligases. Option C, transferases. And option D, lyases. We have discussed the classes of enzyme right now. So I want the answer from you guys. Which among the following type can add water, carbon dioxide, etc. to the double bond? I've told you which enzyme class can add water, ammonia, carbon dioxide to a double bond, or it can also remove these groups to form a double bond. Yeah, Vijayalakshmi has answered option D, lyases. Very good. So option D, lyases is the correct answer. So I've told you the classes of enzymes. So isomerases, what it was doing the isomerization reaction. So basically converting one isomer into the other. Ligases, what it was doing, it was joining two functional group or two molecules with the use of ATP, with the use of energy. Transferases, what it was doing, it was transferring functional groups to acceptor or donor molecules, like to acceptor molecules from donor molecules. Then lyases are basically the enzymes that can add water to the double bond. Okay, so there is no doubt in the classes of enzyme, right? So moving to the next question, question number 10. Enzyme X exhibits maximum activity at pH 6.9. X shows a fairly sharp decrease in its activity when the pH goes much lower than 6.4. One likely interpretation of this pH activity is that a glutamic acid residue on the enzyme is involved in the catalytic reaction. A histidine residue on the enzyme is involved in the catalytic reaction. The enzyme has a metallic cofactor, or the enzyme or the reaction relies on a specific acid-base catalysis. So the activity is going down below 6.4. So that is your actually hint. So I've told you, if I have a reaction whose optimum which has an optimum pH, and if the pH is decreasing, the activity will decrease. But here they have said the activity is sharply decreasing after 6.4. So that means one or the other factor is actually working at that pH. So the correct answer for this question is actually option B. Histidine residue on the enzyme is involved in the catalytic reaction. Why? I have told you the answer. Anybody wants to give the reasoning? So in the previous class, I have told you about pK values of different amino acids. So glutamate is a acidic amino acid. It has an acidic group. So the pK value for that R group is very less. Whereas the pK value of the histidine R group, the side chain of histidine, is around 6. Okay. So if you see, so these are the amino acids that are generally present in the site, like in the active site of the enzymes. Okay, these are the amino acids that are generally present in the active site of the enzyme and can catalyze the enzymatic reactions. Okay, why? Because they have a donor or the acceptor groups that can donate the proton or accept the proton and catalyze a reaction. So basically electron transfer can be facilitated by such groups. Now if you see glutamate or aspartate, they have an acidic group as a proton donor, and if the proton is donated, then as a proton acceptor they have an acidic group. Now this acidic group will work under the acidic pH. So around pH 3-4 these groups can actually work better. So an enzyme that is there in an acidic environment, which enzyme can be there in an acidic environment? So our stomach, our gut pH is our stomach pH is acidic, right? So any enzyme that is present in our stomach that is involved in the digestion of the food, they will be having such amino acids in their active site. Now if you see lysine, arginine, these are basic amino acids. Okay, so basic amino acids, their pK is higher than 7. Okay, and they also have a donor or acceptor group, so they can work in the active site of the enzyme which has a optimum pH at towards the basic range. So based on the groups, based on the side chains, we will see what is the donor or the acceptor group, and if the donor or the acceptor group has the pK value around that particular pH which is asked in the question, we can say okay, this particular amino acid might be there in the active site. So in this question we had the pH 6.4. okay, so the enzyme was active around that pH. So which of the following groups has the pK around that pH? Histidine. So histidine has the pK value for this group around 6. So it can be there in the active site of the enzyme, and that thus the enzyme is functional when the pH is around 6, and the enzyme is non-functional when the pH is dropped below 6. Okay, this is clear? Any doubt in any of this question? Okay, so moving on. Question number 11. The double reciprocal form of the Michaelis-Menten equation, also called the Lineweaver-Burke plot, is given by 1 upon V, that is the rate of the reaction. So reciprocal of the rate of the reaction, 1 upon V is equal to Km upon V max into 1 by substrate concentration S plus 1 by V max. To determine the Km from the double reciprocal plot, you would option A, multiply the reciprocal of the x-axis intercept by -1. Option B, multiply the reciprocal of the y-axis intercept by -1. Option C, take the reciprocal of the y-axis intercept. And option D, take the x-axis intercept where V0 is equal to half of V max. So I've shown you the Lineweaver-Burke plot for the inhibition kinetics. So if you remember the plot, can anybody answer? What could be the value of Km? Okay. So if you see, this is the Lineweaver-Burke plot. So in a Lineweaver-Burke plot, this is basically the reciprocal of Michaelis-Menten equation. So we have the Michaelis-Menten equation as, I'll write here, V is equal to V max multiplied by S divided by Km plus S, right? So if I invert this equation, what will happen? 1 by V is equal to Km upon V max S plus S upon, plus S upon V max into S. So this S and S will get cancelled out, and we will get Km upon V max multiplied by Km upon V max multiplied by 1 by S plus 1 by V max. that is the equation given. Okay, so this is nothing but the reverse inverted reaction, reciprocal reaction, reciprocal equation of this Michaelis-Menten equation. So reciprocal of Michaelis-Menten equation, that is the Line-Weaver-Burke plot. So if you plot, so if you see this equation, you will see 1 by V. If I write it in a linear equation form, so what is linear equation? Y is equal to mx plus c. If I say y is equal to mx plus c, that means I have a y axis, I have the x axis, and I have a line. So the slope of this line is m. So this is x axis and this is y axis, and c is the y intercept. So this point is c. Where the line is crossing the y axis is the y intercept. So this point is c. So now in the Line-Weaver-Burke plot, if this factor is y, so 1 by v, so this axis is, y axis is shown by 1 by v, and 1 by s, this is x. So the x axis is shown as 1 by s. So we have, what will be the slope then? The slope will be Km upon Vmax. So I told you in the previous, when we were discussing the inhibition kinetics from Line-Weaver-Burke plot, Km upon Vmax is nothing but the slope. And what will be the y-intercept? Y-intercept is the c. What is the value of c? 1 upon Vmax. So if I have to find the value of Vmax, I will basically take the reciprocal of the y-intercept to find the value of Vmax. Now what will be the x-intercept? How did they get the x-intercept as minus 1 by km? So to find the x-intercept, we will take y is equal to 0. So wherever the value of y is 0, at that point the line will cut the x-axis, right? So at y is equal to 0, mx will be equal to minus c. What is mx here? Km upon Vmax into 1 upon substrate concentration is equal to minus 1 by Vmax, right? This is right. So if I cancel this out, we will have the x-intercept, that is 1 by s is equal to minus 1 by km. So this is the value they have got here, minus 1 by km. So to calculate the km from a Line-Weaver-Burke plot, we will have to multiply the reciprocal of the x-intercept by minus 1. Why minus 1? Because it is in the negative sign. So to take it in the positive sign. So if I have a value of x-intercept as minus 3, km will be basically equal to 1 by 3. Okay. This is clear. So to determine km from a double reciprocal plot, so this plot is called double reciprocal plot. To determine km from this plot, we will have to multiply the reciprocal of the x-intercept with minus 1. Okay. I hope this is clear. Similarly, if I would have asked how to calculate the Vmax, it will be basically reciprocal of the y-intercept. So if I have y-intercept given here as 1, the Vmax will be equal to 1 by 1, that is equivalent to 1. Or if the y-intercept is given as 2, let's say this value was 2, then Vmax will be equal to 1 by 2, that is 0.5. Okay. This is clear. So the correct answer is multiply the reciprocal of x-axis intercept by 1, that is option A. Moving to the next question, question number 12. An enzyme catalyzes a reaction at a velocity of 20 micromole per minute when the concentration of substrate is 0.01 molar. The Km for this substrate is 1 into 10 raised to the power 5, 10 raised to the power minus 5 molar. Assuming that Michaelis-Menten kinetics are followed, what will be the reaction velocity when the concentration of S is A 1 into 10 raised to the power minus 5 molar and B 1 into 10 raised to the power minus 6 molar. Okay. So reaction velocity is given at a substrate concentration of 0.01 molar, that is 20 micromole per minute, and the Km is given. We have to find the velocity when the substrate concentration is 10 raised to the power minus 5 and 10 raised to the power minus 6 molar. Okay. So we can use the Michaelis-Menten equation, but the question is what is the value of Vmax? So we know the Michaelis-Menten equation is V is equal to Vmax S upon Km plus S. So let's see how to solve this question. So here we have the velocity given as 20 micromole per minute for the substrate concentration of 0.1, right? So this 20 micromole per minute velocity is basically the maximum velocity. Why? Because if you remember, I have told you when S is very higher than Km, V is equal to V max. Okay. Now here the substrate concentration is 0.01 molar and the Km is 10 raised to the power minus 5 molar. So this is basically 10 raised to the power minus 3 molar. Okay. Substrate concentration is 10 raised to the power minus 3 molar and Km is 10 raised to the power minus 5 molar. So 100 times higher than the Km. So S is very higher than the Km. So velocity will be equal to the maximum velocity. So this velocity that is given in the question is nothing but the maximum velocity. Okay. So if I have told you this, so by now you should be able to solve this question. So you have V is equal to V max S upon Km plus S. This is the Michaelis-Menten equation. So when S is equal to Km, so I have told you we have three situations. We have already talked about S is less than Km, S is higher than Km. Now the third situation is S is equal to Km. So when S is equal to Km, so all the terms where S is there in this equation, replace it with Km. So we will have V max Km divided by Km plus Km. So this will be 2 Km and then Km and Km will get cancelled out. So this will be basically V max Km divided by 2 Km. So Km and Km can get cancelled out and V will be equal to V max by 2. So when S is equal to Km, we have V is equal to V max half of V max. Okay. So sometimes you can define Km in terms of substrate concentration also. So Km is that particular substrate concentration when the velocity will be half of the maximum velocity. So if I have a Michaelis-Menten curve, okay, so let's say this is the Vmax. Okay, this is the Vmax. So somewhere here we will have the half Vmax. Okay, so when the velocity is half of the maximum velocity, at that particular velocity, whatever the substrate concentration will be, this particular substrate concentration is nothing but the Km. Okay, so from the Michaelis-Menten equation, from the Michaelis-Menten graph itself, you can determine the value of Km. Okay, so once you have the value of Km, so what is the value of Km? That is 10 raised power minus 5, right? And the substrate concentration is equal to the Km, so velocity will be half of the maximum velocity. Maximum velocity is given as 20 micromole per minute, so half of that is 10 micromole per minute. So option first part of the question is, what will be the reaction velocity when concentration is 10 raised power minus 5 molar? So the velocity will be 10 micromole per minute. So we have two options with that same answer. So A and C. So answer should be between A and C. Let's see. Now the second part is when the substrate concentration is 10 raised power minus 6. So for this, now we can just replace all the values in the Michaelis-Menten equation. So we have V is equal to V max, that is 20 micromole per minute, multiplied by 10 raised power minus 6 molar, that is the substrate concentration, divided by Km, that is 10 raised power minus 5 molar, plus 10 raised power minus 6 molar, that is the substrate concentration. If you solve this whole thing, you will get the answer to be 1.8 micromole per minute. Okay, and this is the answer. This is the reaction rate when the substrate concentration is 10 raised power minus 6 molar. So option A is the correct answer. We have 10 micromole per minute reaction rate when the substrate concentration is equal to Km, and 1.8 micromole per minute when the substrate concentration is 10 raised power minus 6 molar. Okay, this is clear. So these kind of numericals are very common in enzyme kinetics, and you should at least remember the Michaelis-Menten equation. If you remember that, you can solve any question. Even if it is a question of Lineweaver-Burke plot, you just have to reverse the equation and you will get the equation for Lineweaver-Burke, and you can solve the values whatever has been asked. So you just have to understand how they are getting to the like achieving those particular values. So for Lineweaver-Burke, I have explained how they are taking the reciprocal and it is similar to a linear equation. Then you just replace the values from a linear equation. In case of Michaelis-Menten, just remember the basic formula for Michaelis-Menten equation that is given. If you remember that formula, you will be able to solve most of the numericals for enzyme kinetics. Okay. So any doubt in this question? Okay, so we'll move on. Sorry. Question number 13. For a reaction that can take place with or without catalysis by an enzyme, what would be the effect of the enzyme on the standard free energy change of the reaction, activation energy of the reaction, and initial velocity of the reaction? So what would be the effect of the enzyme on these three factors: standard free energy, activation energy, and initial velocity? Anyone can answer? What would be the effect of an enzyme on the standard free energy, activation energy, and initial velocity of a reaction? So let's see what happens in an enzyme kinetic reaction. So whenever there is an enzyme catalyzed reaction, so let's say these are the reactants, that is the substrate, okay, and we have the product here. So there will be some energy, that is potential energy of the substrate or the reactants, okay. So this will be the energy of particular reactants. If there are two reactants, then there will be two like the total energy of the two reactants will be considered. So energy of the reactant, and for a thermodynamic reaction to occur, the energy of the reactant will be higher than the energy of the product. Then only the reaction will move forward, because the reactants will go from a higher energy and reach to a lower energy for a thermodynamic. So this is called a thermodynamically favorable reaction, okay. Okay, so this is the energy of the reactant and this is the energy of the product. In normal case, when there is no enzyme, we will have a reaction curve where the reactants will first reach a higher energy, where the reactants and products will make a complex. So let's say the enzyme substrate complex will be at some higher energy, and then it will reach the lower energy where the enzyme will be released and product will form. So that will have a lower energy than the energy of the reactant itself, okay. So the difference between the energy of the reactant and the energy of the product, that is this delta G, this is called the standard free energy, okay. Okay, so this is called the standard free energy of the reaction, energy of the reactants minus the energy of the product, okay. So this is the standard free energy. What is the activation energy? Activation energy is basically the amount of energy required by the reactants to reach that intermediate state. So this is the intermediate state. So amount of energy that is required by the reactants to reach that intermediate state, this is called the activation energy, okay. So what happens in an enzyme catalyzed reaction? What happens in an enzyme catalyzed reaction is basically, this activation energy is lowered by an enzyme. So when we are adding an enzyme, what the enzyme does is it lowers the activation energy. So the energy of the reactants and products will remain the same, okay. So if the energy of the reactants and products are remaining the same, the standard free energy will be same. There will be no change in the standard free energy. That is clear, because we are not changing the reactants, we are not changing the product, and even the path of the reaction, the path that is the that is taken by the reactants to get converted into product, that is changed by the enzyme. So when we are adding the enzyme, it is taking a shorter route to reach the product, but in the similar manner. So it is again reaching an intermediate, but now in this case, this enzyme substrate complex is made at a lower energy than what it was making when there was no enzyme, okay. So if we have here the intermediate made at a lower energy, that means the activation energy is now lowered when an enzyme is present. So we had the option standard free energy change. So for one, there will be no change. There will be no change in the standard free energy. Why? Because the product and the reactants are same. So the difference between the energy of the reactant and the product will also be the same, okay. So the standard free energy will be same irrespective of the enzyme being used or not used, okay. So if an enzyme is added, there will be no change in the standard free energy of the reaction. There will be a change in the activation energy. What is that change? The activation energy is reduced or decreased. Okay, that is what the enzyme is doing. Why the enzyme is catalyzing the reaction faster? It is called a catalyst. If we are adding an enzyme to a reaction, the reaction is happening at a much faster rate than that particular reaction would have happened if there was no enzyme present, right? So that's why the enzyme is taking a shorter route to reach to the product. So that's why the enzyme is catalyzing this reaction faster. So the enzyme is lowering the activation energy. By lowering the activation energy, it is reaching that intermediate state at a faster rate than the reaction was going without an enzyme, and thus it is taking lesser time to catalyze the reaction. And then the initial velocity of the reaction. So if I am adding an enzyme, I told you the enzyme is going faster. So if the enzyme is going faster, what is happening to the velocity? It is increasing. So initial velocity of the reaction has increased. So there is no change in the standard free energy. There's a decrease in the activation energy, and there's an increase in the initial velocity of the reaction. Okay, so option A is the correct answer. I hope this is clear to everyone. So there are three terms: standard free energy, activation energy, and then velocity. So I have explained why the standard free energy is not changing. Why it is not changing? Because the reactants and the products are same. So the difference in the energy between the reactants and the product will also be the same. Activation energy, it is decreased when an enzyme is added, so that a shorter route can be taken to reach the product faster. Since the enzyme after the addition of enzyme, the reaction is proceeding at a much faster rate, that means the velocity has increased. Okay, this is clear? Any doubt? Okay, if there's no more doubt, we will move to the next question. Question number 14. A small molecule that decreases the activity of an enzyme by binding to a site other than the catalytic site is termed as option A, allosteric inhibitor, option B, alternative inhibitor, option C, competitive inhibitor, and option D, stereospecific agent. A small molecule that decreases the activity of an enzyme by binding to a site other than the catalytic site is termed as. Anybody knows? Okay, Karuna has answered option C. Anyone else? Come on guys, answer. This is supposed to be an interactive session, so it would be great if you guys answer from your end, and then we can discuss why the answer is correct or wrong. This will actually help you understand better. Even if you know the wrong answer, it's okay to answer. Anyone else wants to answer? Okay, so let's see. The correct answer is actually allosteric inhibitor, that is option A. So what is the difference between competitive and allosteric inhibitor? I'll explain. So I told you an enzyme has a catalytic site where the substrate can sit, bind to the enzyme, and then get converted into product, right? Now a competitive inhibitor will have a similar structure to the substrate. Okay, let's say this is a competitive inhibitor. So this will have the similar structure that can fit in the catalytic site of the enzyme. So if the catalytic site of the enzyme is made like this, and we have a substrate that sits here. Okay, so this is the enzyme, this is the substrate, and now I make an inhibitor, a small molecule inhibitor like this. This can also, since the site is similar to the pocket of the enzyme, This inhibitor can also fit, and now I add higher concentration of this inhibitor. So inhibitor concentration is higher than the substrate concentration. In that case, what will happen? The inhibitor will compete for the binding with the substrate. So inhibitor and substrate will compete. Compete for what? Compete for binding to the enzyme. So if a substrate binds to the enzyme, it will get converted into product. But if an inhibitor binds to the enzyme, it will block the enzyme. Since that inhibitor cannot get converted into product, so that will block the enzyme. Once the enzyme is blocked, substrate cannot come and bind. Okay, so one site is being competed by two molecules, that is competitive inhibitor. What is happening in case of allosteric inhibitor? So this is an example of competitive inhibitor. Now what is happening in case of allosteric inhibitor? Just a second. So what is happening in case of allosteric inhibitor? So we have the enzyme and we have two pockets in the enzyme. Okay, so this is the main catalytic pocket where the substrate can bind. So this is the substrate, this is the enzyme, and it has another small pocket away from the active site. Okay, another small pocket away from the active site where another small molecule can bind. If this molecule is binding to this pocket, it can actually inhibit the reaction of the substrate converted into product. So this is called an allosteric inhibition, a small molecule that decreases the activity of the enzyme by binding to a site other than the catalytic site. So this is a site which is away from the catalytic site. It is not a part of the catalytic site, and by binding to that particular site, what it does is basically, so let's say we have the enzyme to which at an allosteric position, now the molecule has bound. Once the molecule has bound to that allosteric position, it kind of deforms the catalytic site. Okay, and the catalytic site, since it is deformed, now it is not in this triangular shape, the substrate cannot fit in this pocket. Now if the substrate cannot fit in this pocket, the catalysis won't happen and the product conversion will be inhibited. This is clear? This is clear? So this is called an allosteric inhibitor. So the examples of inhibition kinetics that we studied in non-competitive or uncompetitive inhibition is actually due to these allosteric molecules. Okay, the molecules that can bind to the site other than the catalytic site and cause the inhibition. Okay, I hope this is clear. So the difference between competitive and the allosteric inhibitor is clear, I hope. So moving to the next question. Based on the requirement of activation energy, enzymes can be defined as those which do not require activation energy, lower the requirement of activation energy, increase the requirement of activation energy, and do not change the requirement of activation energy. I've just now explained this question, so I want the answer from you guys. I'm not giving the answer for this question. So tell me, what is the case? What does an enzyme do to the activation energy? Yes, Vijay Lakshmi has answered option B. Lowers the requirement of activation energy. Very good. So I've shown you the energy diagram right now. So you all have seen the energy diagram. I'll show it again. So it is basically decreasing the activation energy. Okay. So there should not be any doubt in this question, I hope. So option B, lower the requirement of activation energy. Now next question is question number 16. A certain enzyme will hydrolyze egg white but it won't hydrolyze the starch. Which statement best explains this observation? Option A, starch molecules are too large to be hydrolyzed. Option B, enzyme molecules are specific in their actions. Option C, egg white acts as a coenzyme for hydrolysis. And option D, starch is composed of amino acids. So which of the following is true? Why one particular enzyme will hydrolyze egg white but not starch? Yes, again Vijayalakshmi has answered. Very good. Option B. So enzyme molecules are specific in their action. So I have just told you about the catalytic site. So each enzyme has a particular shape of catalytic site which can fit only one type of enzyme. Okay, so one type of structure. So it's called a key lock and key mechanism. Okay, so you know each lock has one type of key that can fit in that lock, right? You can't open any lock with any key, right? Similarly, you cannot have any substrate for any enzyme. So one enzyme will bind to one class of substrate. Okay, so if a enzyme, like I'm just drawing a rough cartoon, that if an enzyme catalytic site has this shape, all the substrates that will have this shape can fit in this pocket. If the shape of the catalytic site is star like or something, then only substrates that will have that particular conformation will fit in the pocket. So any substrate that is fitting into the enzyme pocket will only get catalyzed by that particular enzyme. Okay, so every enzyme is very specific in their action, and because of this specificity we have so many functions going on in our body with different types of enzyme in a single cell. So in a single compartment you can have hundreds and thousands of enzyme, and each enzyme will know which substrate it will bind to and catalyze the reaction. It won't happen, it will never happen like I have some enzyme and I am giving some other substrate, and since the enzyme is not finding its native substrate, it will start catalyzing the other substrate. It will never happen. Okay, so each enzyme is very specific. So if I'm giving the enzyme, if the native substrate of enzyme is egg white and I'm providing the enzyme with starch, it will never hydrolyze the starch, although it knows how to hydrolyze. It knows the process of hydrolysis, it knows how to break a bond and add water to it, but it will never hydrolyze starch or any other molecule. It can only and only hydrolyze egg white. Okay, so that is called a specificity of an enzyme molecule. And the proposed mechanism for the specificity, one of the proposed mechanism is lock and key mechanism. There's another proposed mechanism, that is the induced fit model, which is also giving a hypothesis how the enzyme has this particular specificity towards its substrate molecules. Okay, so option B is the correct answer. Enzyme molecules are specific in their actions. Okay, moving to the next question, question number 17. Calculate the ratio of substrate concentrations required when the reaction rate is 90% of Vmax to the substrate required for 10% of Vmax. So we have to calculate the ratio between the reaction rate. So rate of reaction when the we have to calculate the ratio of the substrate concentrations. Okay, so we have to see when the rate of reaction is 90% of Vmax, what is the substrate concentration coming from the Michaelis-Menten equation. We can solve this question. Okay, what is the Michaelis-Menten equation? V is equal to Vmax S upon Km plus S. Okay, now we have to calculate for S when the V is equal to 90% of Vmax, and in second case we will calculate for S when V is equal to 10% of Vmax, and then we will take the ratio of those two particular substrate concentration and we will see what is the ratio coming. Okay. So let's see how to solve this. Just a second. So we have the Michaelis-Menten equation, V is equal to Vmax S plus Vmax S upon Km plus S. Okay. Now we are saying V is equal to 90% of Vmax. That is, we can write V is equal to 0.9 Vmax. Why 0.9? So 90% means 90 divided by 100. So 0.9 Vmax. So we will just replace the value of V with 0.9 Vmax in one case and 0.1 Vmax in other case. So when we have 0.9 Vmax, in that case we are calling the substrate that particular substrate concentration as S with 0.9 in the subscript. Okay. So substrate concentration when rate of the reaction is 90% of Vmax, if we calculate, it is coming to 9 km. So 0.9 Vmax is equal to Vmax S upon Km plus S. So we can cancel out the Vmax here and we will get 0.9 km. So you can carry the denominator here. So 0.9 km plus 0.9 S is equal to S. So if you solve this further, you will get km 9 km is equal to S. So S is equal to 9 km when V is equal to 90% of Vmax. Now in the second case we were asked V is equal to 10% of Vmax. So 10% means 0.1 Vmax. Again solve for Michaelis-Menten equation. So Vmax gets cancelled out and then rest, if you carry the denominator on the other side of the equation and then solve for the equation, you will get S is equal to 1 by 9 km. Now we were asked the ratio of the substrate concentration at V of 90% of Vmax and substrate concentration when the rate was 10% of Vmax. Okay. So S 0.9 divided by S 0.1 is equal to 9 km divided by 1 by 9 km. So the ratio comes to 81. So km will get cancelled out by km and we will get the ratio to be 81. Now the interesting thing about this question is, for each and every reaction that is following Michaelis-Menten equation, the ratio between two different reactions where the velocity is a part, like velocity will always be a part of Vmax, right? So the ratio between two reactions, ratio of the substrate concentration between two reaction will always be a constant, irrespective of the km value, irrespective of the particular substrate concentration that you have taken. If the reaction is following Michaelis-Menten equation, the ratio of the substrate concentrations for two different reactions will always be constant. Okay. So here the answer is 81, that is option number C. So this is clear. So if I had, instead of 10%, if I had 50% of Vmax, so only this factor would have changed to 0.5 Vmax and the rest of the things would have been same. And if you would have solved for this, you would have gotten S is equal to Km, which is actually true when Vmax is, so when velocity is half of Vmax, we have S is equal to Km, right? And in that case, you can again solve for the ratio of S 0.9 by S 0.5, and that particular ratio will always be the same irrespective of which enzyme you are taking, what is the Km of the enzyme. The Michaelis-Menten equation is constant, so the ratio of two substrate concentrations will always be a constant. Okay, I hope this is clear. Any doubt? So we are moving to the last question of the session. Question number 18. An enzyme was assayed at an initial substrate concentration of 2 into 10 raised power minus 5 molar. In 6 minutes, half of the substrate has been used. The Km of the substrate is 5 into 10 raised power minus 3 molar. The value of K, that is the rate constant in the unit second inverse, is. So the unit is mentioned. Why the unit is mentioned, we will see. So first of all, let's see how to calculate for this rate constant. So if you now first you have to see the value of substrate concentration and the Km. Can you fit it in any of the three cases that I have told you, where the substrate concentration is very high than Km, substrate concentration is very low than Km, and the substrate concentration is equal to Km. So in this case, if you see, the substrate concentration is very lower than the Km value. The substrate concentration is almost 100 times lesser than the Km value, right? So if that is the case, I have told you what is the order of the reaction. The order is first order reaction. So let's see how to solve. So when the S is very low than Km, when very lower than the Km, we have the reaction as the first order reaction where V is directly proportional to S. And this Vmax and Km, these two are constant entities. So Vmax by Km, this ratio could be written as K, that is the rate constant of the, since it's a first order reaction, it will have a rate constant. Okay, it is the rate constant of that particular first order reaction, of the overall reaction, overall enzyme reaction that is going on. Okay, and it is, rate can also be given as the amount of substrate concentration that is consumed in the reaction per unit time. So minus ds by dt. Why minus? Because substrate is being reduced, like substrate is being used. Now if you derive the equation, so I am not going through the derivation completely. If you derive the equation, you will find a linear equation in terms of log, and here we have a statement, in 6 minutes half of the substrate has been used. What does this mean? That means the half-life of the enzyme. What do you mean by half-life of an enzyme? Half-life of any molecule means the time taken for the half reaction to complete. So if I have a substrate, if I have an enzyme and a substrate, the time taken for that enzyme to catalyze half of the substrate into product is the half-life of that enzyme. Okay, so T half is basically when the substrate concentration, the initial substrate concentration, the final substrate concentration is half of the initial substrate concentration. Okay, and if you see the derivation, you will find the T half can be written as 0.693 divided by K. So now in the question we have the T half given, that is the half-life given as 6 minutes. Okay, so if I have the half-life as 6 minutes, we have to solve for K. We can directly solve for K by dividing it by, say, 0.693. So 0.693 divided by 6 minutes will give you the half-life, but this will give you the rate constant value, but this rate constant value will be in per minute because the unit of time is given in minute. Okay, now in the question it is specifically given that that the answer has to be given in second inverse. So you have to see the unit that is given in the option also. Okay, so 0.115 is also an option. So once you get this answer, do not take that answer because you have to see the unit also. Now the unit is in minutes, so we have to convert it into second. So divide it by 60 again, and then you will get the final answer, that is 1.925 into 10 raised power minus 3 per second. That is option number C. That is the correct answer. Okay. This is also one of the important questions in this assignment. Any doubt in any of the question? This was the last question of this assignment. So any doubt in any of the questions? We have 5 minutes left, so we can discuss if anyone has any doubt. If any of you has any doubt in any of the previous questions also, we can discuss, okay? So in this whole session, the most important thing is to remember the Michaelis-Menten equation, because most of the questions could be solved based on that equation itself, okay? Understand the concept of the binding of the substrate molecule to an enzyme. What is the Km, Kcat? These terms signify, what does these terms signify? The catalytic efficiency of enzyme, how it is affecting the reaction. So these are some of the concepts that you have to understand completely in order to understand the enzyme kinetics, okay? So if you don't have any doubt, then we can maybe end this meeting. I'll wait for 2 more minutes and then we can. So I'm just going through all the slides. If anyone remembers anything they did not ask or anything, you can ask. So this is actually a very important chapter. If you are going to do biochemistry in future also, if you are going to design experiments, this is a very important chapter. So go through the concepts very nicely and try to understand as much as you can. Okay, the recording of the session will be uploaded and the lecture videos are already there on the portal. So go through the lecture videos, go through this recording also, try to solve the assignment that has been given to you, and hopefully you will do well. Okay, then if there's no more doubt, we can end this session now. Thank you so much for joining.